right, we're getting close to starting. All right, looks like we have about 32 people. I'm going to wait another couple minutes in case people are having any trouble logging in. Check my email in case anybody is saying they're having trouble. Okay, we're going to try something in the lecture today. In case I need to draw anything, I'm going to try and share this, this whiteboard. So let's see if this, I'm pasting a link to everybody in the chat. We may not need to use it, but if we do, let's see if that works. And Tanya, you should also have access to it. Not that you should draw over me, but I think you can. I think you'll be able to get onto it as well.
Sure.
Assuming my iPad stays charged. Okay. All right. All right. I'm gonna get. Let's give everybody another minute or two. Okay, I'm going to post a poll in just a second if you want to give some feedback on things that you might want to hear more about, and then I can figure out how to unmute folks that want to ask questions as we get to it. I'm just going to do a poll. You can vote. I mean, I'll probably do them all, but let's see. Okay. All right, so I posted a poll for folks who want to just think about, kind of prompt you for a couple of different ideas about just big topics that we talked about in the lecture, and see if you have any feedback about that, and I can start. I can start going over specifics. I think. Let's see if you. I think everyone is muted by default, but I think you can unmute yourself. Do you have permission to do that? Because I say, Tanya, can you unmute yourself? Yeah.
Yeah, I can unmute.
Okay. And so, welcome back to Lecture 2, or the Q&A for Lecture 2. We're going to go ahead and get started, and then I can obviously circle back around if folks come in late and have some more questions. But today's lecture, or this week's lecture, second lecture, was on sequencing technologies and an introduction to kind of overview of the genome, and a little bit about how we've estimated genome complexity and size in the past, and a beginning on how the sequencing technologies work, the chemistry behind those. So hopefully you got a view of that from your from watching the lecture or reading the slides. And so I guess I'm happy to sort of go back over aspects of that, and for parts that didn't make sense or parts you'd like more explanation. The poll is kind of coming in. People are asking for most of these topics to kind of be recovered, which is great. But it might help if anybody has a specific part of that you would like a little bit more input on. But I can certainly start with the sequencing technology as one topic, and then jump in further. So if somebody wants to unmute themselves and ask the question, that's great. Ask a question, or if you want to write something out that wasn't clear, if there's a particular slide that that you would like me to go over more, maybe help direct me on that. Okay. Let's see. Great. Okay, I got two questions. Okay, let me go 2 questions about the ploidy and depth of coverage. So that's great. Let's start with that, and then we can come back to technologies again. All right. So I'm going to share my screen, and then if need be, I can draw on that link that I've posted as well. But let's see if this makes sense. Okay. Let's make this big. Okay, looks good. You can see my full screen, yeah? Hope. Let me know if you can't. Tanya, you're going to tell me if you can't see the screen, right? It's good. Okay, good. Thanks. All right. So let's talk about the sliding window concept first, and then we can talk about how we use this to kind of understand ploidy. First of all, the idea about the sliding window analysis, or the depth of coverage, is that we are sequencing the genome of an organism, right? So we've taken the DNA of that organism and we've chopped it up in small pieces, and we can only sequence small fragments, right? So even though the genome is 3 billion bases or even just 1 million bases, we can't sequence all of it at one time with a lot of these technologies. And so what we're doing is sequencing a small bit of it. Now, in this particular example, when we're talking about aneuploidy or some aspect of ploidy, we know the genome already, okay? So we kind of jumped ahead and said, guess what? We already know the genome, but we're resequencing another individual. And what we're doing is we're taking that individual, we're getting its DNA out of the cells, and we're sequencing in a technology, typically the Illumina technology, which I talk about later on in this lecture. So we get these small fragments, and what we do is we align those fragments. We basically look for where that small piece of DNA came from in the genome. You can kind of think about it like a search and the find command in like Word, right? You've basically got a set of sequences, a set of base pairs, let's say 100 base pairs, and you've asked a program to go in and figure out, well, where did this 100 base pairs come from in the genome? And so we've lined that up, and that's what these little lines here on the top here are drawing, are the is a representation of that piece of genome, where it came from in the genome, okay? And so remember, the process is random, right? We just take cells, we grind them up, we take that DNA out, and we sequence it, and that's random. So we're just sampling 100 base pairs at a time randomly from the genome. So on average, if we did that a lot of time, all places across the genome, we would have sampled every bit of the DNA in the genome, assuming all of it could be sequenced. And so if we do that over time, or over a lot of samples, sorry, a lot of sequence, then we would start to put these sequences down, and they would be piled up kind of graphically the way we're drawing them here, as if we have a little bit of the genome that we've sampled. And so kind of that's the first step in these analyses, is to basically say, where did this little piece of DNA that I've sequenced come from in the bigger genome? Okay, now that I have that, I can kind of write down in a like a big computer program and basically a big data file, where did that little piece come from? So I do that over and over again, and I have a big map of where all these pieces came from. And again, if I then go down to every single base in the genome and I ask how many bases, how many sequences overlapped with that one base. So if you think about in this picture here, there's a box, and so I'm going to ask for that box, which let's say it's 100, it's 1,000 bases long. I'm asking how many sequences fall into that box, and that becomes a number, and that number is the depth of coverage. That's how much sequence did I sequence in that for that spot. Okay, and again, if everything was kind of equal, then we would expect to see about the same depth of coverage across the entire genome, everywhere we go, because it's a random sample. We should on average be able to sequence all of the genome, so we'd have a random depth, average depth. Okay, but there are places in the genome which when we compare different individuals might be missing. So if you had something that was missing, there would be no reads for that region in the genome, and the value would be 0 for the depth of coverage. And in other cases, we have things where the coverage is higher, and we have extra copies of something. And so that's where you can see these kind of pictures where it starts to get higher. So the top part is kind of representation of where the sequences came from in the genome and the sequencing that we did. And then the bottom part is how we would, it's kind of the graphical view of how we would look at the depth of coverage and compute it. So we do this across an entire genome, and that's what this sort of sliding window is supposed to show. We're going to calculate it for every single window. How deep, how many sequences were there? How many sequences did I recover in my sequencing run for that region of the genome? So that's the sliding depth of window coverages. And here's a picture of an actual data. So every point is a computed depth of coverage at a particular spot in the genome. So again, you can see on average, it's kind of around 1, because we've done a normalization so that the average basically is 1. But you can see in general, it's a little bit wiggly because some places seem to have a little bit more than others. And that's just different aspects of the chemistry and the randomness kind of come into that. But that's what you see if you took every single window across, in this case, 1 chromosome, and asked what's the average depth of coverage of sequencing for that spot. Okay, let me stop there. So Does that help, Eric? Are you still confused about the idea of what that sliding window is doing? It's okay to unmute and ask the question if you guys are. Okay, oh good, Eric said that helps a lot, great. Okay, so then the next component of this I started, so we will talk a little bit more about like the methods behind doing this, but I wanted you to see kind of what data look like when we process it, when we actually start to use sequences. So the other aspect to this was this idea about ploidy. And so this is a really simple view of this where we can just scan along the genome and ask where is that average coverage different? And you can see in this case that chromosome 13 has an average depth of coverage that's a bit as higher than the rest of the genome. And that's what an aneuploidy would look like. If it's above the line, like, so again, an average being 1, if it's above that, that means there's an extra copy. And if there was a region of the genome or a chromosome that didn't have anything, that was missing, then that would be a case where we had an aneuploidy and we lost a chromosome. So that's what this really hopefully simple to understand idea of the sliding window analysis, without any kind of fancy statistics. It's really just calculating this sort of average depth of coverage in a window can tell us whether or not a chromosome is gained or missing in an aneuploidy. Okay, so then I took it to another level, which was to say that we could think about this not just at a whole chromosome level, but we can also estimate this using what's called allele frequencies. And we'll talk about this a little bit more as we do analyses of genetic variants, but this also works for ploidy. And so the idea here is that we have that we have a marker. In this case, we're just talking about one place in the genome, and we have a marker where there's a difference between a position in the genome, and in general, we will have some reference genome, right? So like for human, there's multiple reference genomes, but there's some space, some position in the DNA where we have an A and the reference has a C, right? And so that's something we can detect with sequencing technologies and bioinformatics. And so all we're doing is we're just counting how many times we see a C or an A at that spot. Now I've simplified it even more. I've made it red and gray, but like, so at a particular spot in the genome, there's a red spot, there's either a red or gray allele. And so when we look at allele frequencies, we kind of have some expectations that if the organism is haploid, we're either going to see an A or a C there 100% of the time, right? Because there's, at that particular place in the genome, there should only be one chromosome. In this case, there's only one chromosome in the organism, but there should only be one kind of copy of that chromosome. However, if it's a diploid, there could be 2 alleles that exist there. And again, maybe you remember this when you talk about heterozygosity in introductory genetics, right? And you have a big A and a little a, right? That's what's going on in the n equals 2 here, right? You have a red or a gray. Big A, little a is kind of a way you could think about that too, and that there were 2 alleles at the same same position. And there, if you were to do DNA sequencing, you would find those alleles at 50-50, a 50-50 ratio. You would see 50 A's and 50 C's if you had 100 sequences for there. If you only had 10 sequences, right, you'd had 5 A's and 5 C's. So it's just a ratio or a percentage. But so again, what we would expect in a diploid organism, if we scan along the genome and look at SNPs, so these are single nucleotide polymorphisms, right, positions in the genome that vary within an individual. Right, remember you have in humans, we have 2 copies of a chromosome, right? So therefore, we would always expect to find at a particular place in the genome, when we're looking at a SNP, let's say, so a single base pair, we would always expect the ratio of those alleles to be really close to 50-50, right? There might be a little bit of variation. It might be like 49-51 or even 45-55, right? That percentage could vary a little bit because there's some randomness. But in general, we'd expect it to be at 50-50, okay? If we did this and we scanned along and we found a position in the genome and we counted all the alleles that we saw, and the frequency at a spot was 1/3, right? So you had 33 A's and 66 C's. Then you would be in a human, you'd be very surprised. You go, that's not what I expect. I expect the ratio to be 50-50. So it tells you that in that case, if you don't know the ploidy, it tells you that the ploidy probably isn't 2. The ploidy is probably 3. Okay. Now, if you see something at 25%, again, that would tell you something that it's n equals 4. Now, you can, of course, have alleles which are at 50-50 in a tetraploid where n equals 4, right? Because there are scenarios where there would be 3 different alleles present. The ratio would be 1 to 1. Even though there's 4 chromosomes, because you could of course have that happen, but you have to look at the total collection of allele frequency. So I think on this slide, I was trying to show you again what that would look like. So you might recover two different alleles at a particular position where the arrows are here, right? So you either see an A or you see a T at that position. And all we're doing is we're just counting up the number of times we see an A there and the number of times we see a T there from the sequences that we got that came from that place in the genome. Okay? So if that ratio is 50-50, then we're gonna make an assumption that it's a diploid. Okay? Now, as I said, you could have the case where the allele frequencies are 50-50 in a tetraploid. Okay? So how could we distinguish between those scenarios? And that's why in this other slide, I'm showing you the complete set, the total set of allele frequencies in a histogram. Okay, and so in this left-hand side where we have the one that I've labeled diploid, the CBS 7837, that's that range. So these are from yeast, Saccharomyces cerevisiae, right? The things that make that stuff that makes bread for you, but it is a particular strain of Saccharomyces, which is a diploid. Now, Saccharomyces can have, it's a little bit flexible in what its ploidy can be. So you can actually have Saccharomyces that's triploid and Saccharomyces that's tetraploid. You can manipulate it so it will exist that way. And so all these different strain numbers are strains that have different ploidies. And so if you a priori you didn't know what the ploidy was, if you went across the genome and everywhere where you saw a SNP, right? So where at a particular position in the genome there were 2 different alleles that showed up. Then you just look at the ratio of the number of the frequency of those, and that ratio tells you something about what the ploidy might be. And so if you looked at the total set for all the SNPs in the genome and you looked at that distribution, and you would see most of them really close to 50-50, right? That's what we're seeing here on the left-hand side. And then if we look in the middle one, we see that it's we have 2 peaks, right? One peak at 1/3 and one peak at 2/3, because you either have either of those scenarios depending on whether or not the allele, whether or not there are 2 in this particular example here, right? For the n equals 3, you can either have 1 copy out of 3 or 2 copies out of 3 in order to detect it, right? The reason why I don't show you the case where all 3 chromosomes have a red there is because then that wouldn't be a variant within this individual, right? If every time, for most places in the genome, if everybody agrees on the SNP on that position, then it's not variable. So it's not something we're considering. So we're only considering the case where there's some difference that's showing up. Okay, let me stop there for a second. Does that help, Alexander? Let's see. So Genesis, I guess you asked about the ploidy. Let me know if that makes sense to you. Great. Alexander just mentioned in his question, just does the higher depth of coverage be due to transposons? And so, okay, and you get it. Yeah, so places where we see higher coverage could be a transposon, but we do have cases where not just one little small spot of the genome is high coverage. It might be the whole part of an arm, like a whole arm of a chromosome. So if like an arm broke off, let's say this is a kind of aneuploidy in humans, right? You can lose a part of a chromosome arm. So one of your chromosomes has the whole set, and then the other copy is missing part of it, right? So that would look like here, it would in this kind of plot, you would kind of go along at 1, and then at some point it would drop down to 0, or sorry, yeah, it would drop down to half actually for that, because you'd have half as much as you would expect to see, right? Because we've normalized this to 1 the way it's being drawn. So you'd have half as much DNA at that spot if you were missing one of the arms of a chromosome. So it doesn't have to be localized to just one window. It could be a whole half of a chromosome, or as in this case, you can see that things have gone from 1 to 2, and that's what you see up there. Okay, so again, some methods are more or less sensitive, but this is what we can put up some more papers of this if you would like to see. But this is how people are identifying cancer. Certain kinds of cancers are basically the result of an aneuploidy, where you've either gained a part of a chromosome or you've lost a part of a chromosome, and then that means you have extra copies of genes that might regulate growth, or you might have lost copies of genes that repress growth. And so cancer, of course, is a result of growth that's not being regulated. And so cells just keep dividing and dividing and take up all these resources. And so oftentimes the causes of cancer are related to losing or gaining genes that are related in the pathways that control growth. Okay. Tanya asked if our SNPs related to allele frequency. So SNPs are the units that we're looking for to try to understand their allele frequency. So they are a place in the genome, and we're asking what that differs within an individual, and we're asking what's the ratio of the frequency of that. So I guess I say again, what is the frequency of that particular allele? Is it 50-50, or is it some other other value? And looking at the total set of those allele frequencies can tell us something about what's the likely ploidy of the organism. All right, I hope that maybe filled in some gaps there from what was in the lecture and got you about it in another, think about it again. Does anybody else have questions about ploidy while I'm here on these slides, or things I can re-explain if that's helpful? Cool, okay. I'm happy to go back if somebody realizes something doesn't make sense. Maybe you're getting a chance to re-review the slides while you're listening here. Let me just go a little bit forward. So maybe I'll just briefly cover the genome sequencing technologies again so that you get a sense of that. And I think these are what I want you to understand about sequencing technologies is that they have some advantages and disadvantages. There's a couple of different ones of these, a couple of different technologies, and they've existed through different periods of science, and they all still are used in some ways, but they have different advantages. And so Sanger sequencing was really, again, the first kind of way that we were able to really effectively sequence kind of large amounts of DNA, and because the technology was relatively efficient. But the important kind of concept here is that Sanger sequencing uses a dye terminator sequencing, which just means that there's this that it basically extends the sequencing is performed by adding a base at a time to some sort of based on some sort of template. So the template is the DNA that came from the organism, or the locus that you're trying to understand, and you're adding one base at a time to that. And then the way that you detect that addition of a base is that there's some sort of fluorescent signal, some sort of fluorophore, or some way that it's being detected. Or in the first iteration of that, what I'm showing you here at the bottom is called a sequencing gel. And again, that's like the original way that folks were able to do it. And the reason why those different bands are on the gel is they represent the different so remember, electrophoresis is that you're running DNA or running something out on a gel that is going to be kind of the bigger the size or the heavier it is, the slower it's going to run. And so you read these sequencing gels from the top down. I mean, from the from the bottom up. You're getting things that are basically that have run the fastest are going to be there in the going to go to the bottom. But basically, you're going to read it in order. So that in the way that it's drawn here, what you can see where it starts with the T, and then there's an A that comes after that, then there's a G, and there's a C. So each of the base pairs was run in a separate lane in this particular gel, and you're able to tell what comes before the other one because they're a little bit different in size. And so it's because if you look at the picture above that, they're all part of this template where you've only added one base at a time. anyways, I don't really need you to understand that much about the chemistry beyond that. I just want you to understand that this approach, this is what existed, and this was one of the first ways we could do it. It's it has advantages in that you can sequence about 700 bases for it. So it's still something that we use in the labs today because it's still relatively inexpensive to do a little bit of sequencing with it. But when you want to sequence millions of bases, it's just not cost effective or time efficient to be able to do it. Even though we don't use these gels anymore, we can do everything through what's called a capillary gel. So basically you run the gel in a really small tube instead of running it on like a rig, and you can read as things pass through that. So I just want you to kind of get a sense that that's the first way it was done. A lot of important science was done with that. But then when a couple of different technologies, I didn't talk about 454, which is another technology, but talking about Illumina here is kind of because this is the dominant technology right now. This is if you were to go to your doctor and get a DNA sequencing test for your genotype, right, and you wanted to see what you might have risk factors for, this is probably the cheapest way it can be done. That they still sometimes, some of those technologies still do what's called exon capture, where they only sequence the genes by looking at sequencing your RNA, your messenger RNAs, but it's at the point where it's so cheap that typically you can sequence a whole genome of a human in just a day or two, and about $1,000, I guess. I mean, it's probably about $100 and $200 probably in cost right now, depending on the group that's selling it. But anyways, but the important part, which what we're learning in this class, is data is one, but actually understanding and interpreting the data and analyzing it is the next thing. So that's kind of what we tackle in this class. Kathy's asking about, is there a preferred type of sequencing technology, or does it depend on your sequencing? Great question. Okay, so, and I think that's the essence of what I want you to get out of this, which I hopefully have explained somewhat, but let me cover here, is that these different technologies have different advantages, and we often use them in for different questions. And so Illumina is really, it's inexpensive, and it can produce an incredible amount of data, right? So it has the best cost, the price performance, if you think about it that way, right? It's you can generate a lot of data for not that much money. The disadvantage to it is that what you generate is pretty short. We can only sequence about 200 to 300 bases at a time. We can do a lot of those 300 bases at a time, but we can only sequence a short fragment. And so that means to put everything back together, to make a genome out of those short fragments, we have to do a lot of computational analysis to figure out how they all fit together. Right, so you could imagine if you have a big puzzle and you were to cut up puzzle pieces, right? If you use Illumina as your kind of cutter, you have a lot of really small pieces in your puzzle. Okay, if your puzzle is mostly blue sky and clouds, right? It's sometimes kind of hard to put that back together, right? Different parts of the sky might have the same color blue. And we'll talk about this when we talk about assembly on Tuesday or the next lecture. But that is a problem, right? And so what the blue part is, right, is something that's repetitive, right? I can't tell the difference between the blue in the sky that's on the left-hand side and the right-hand side. I need more context. So Illumina is really short fragments, but lots and lots of those, and actually lots and lots of those, so we can get really deep coverage of a genome. Okay, so it's cheap, it's fast, but it only generates short reads. So that's I guess what I want you to think about in terms of advantages or disadvantages. So we use it for doing a lot of those things I was talking about before, to look at ploidy or to look for SNPs, which is really probably the really big sweet spot for this project, for this technology, right? If we wanted to look in your genome and identify what makes regions of your genome different from the reference genome, or if we knew about a particular SNP or a particular variation that was linked to a disease, that you were predisposed to a disease, or maybe it was linked to certain kinds of P450, I've mentioned this last time, P450 genes that might affect your drug metabolism. So you would want, we might only need to find 1 or 2 bases in your genome around those genes to figure out whether or not you are, or the patient or whoever it is, had different alleles for that at that site, right? Maybe they have an allele that makes them unable to metabolize a certain drug. We want to know that, right? So Illumina is really good for that because we can find those small places where that is. What Illumina doesn't do well on is the idea that we might have a larger region that is really different. And it's harder to deal with that because the sequencing is so short from Illumina. Okay, so, and as we do a little bit more with alignments, this will maybe be clearer too, but that's the aspect of Illumina. And then we think about PacBio, which is another technology. There's sort of 2 other technologies that produce longer reads. So PacBio and Nanopore, right? And they're kind of competitors. They both have, both these companies, both these technologies are really changing the way that we can do sequencing because they can generate really long fragments. The technology underneath them is different. PacBio uses essentially a biological system. It uses a polymerase, right? Which we have polymerases, all living organisms have polymerases or need polymerases to replicate their DNA, or you have, that's a DNA polymerase, or you can have an RNA polymerase, which makes RNA from DNA, right? So you've got these different sort of cellular machines. And what the folks that first, or the scientists that went and started doing this research and figured out a way to kind of co-opt is they made a special version of the polymerase that can, that stays in place. So they've engineered it so it has this ability to be sort of stuck in the bottom of these little wells. And you can see 100 nanometers at these little wells. And then they've designed the machines and this whole approach to be able to push, to put a DNA fragment into those wells and to let the polymerase replicate it and process it. And as it does it, it doesn't just make another copy of it, it makes a copy and it generates a signal. It generates a light, a detectable light basically emitting. And so that allows, this fluorescence pulse is what gets detected, and that allows, if you read a bunch of those pulses, you can figure out what were the nucleotides that were being integrated as it went through this replication. So it's kind of like, they call it a movie because it's like you're like recording how the polymerase is doing its job, which is super cool, right? Like you've taken this like molecular machine and you've kind of co-opted it and using it to do it. And so they didn't have to invent anything new in terms of how to make replication work. There already existed a molecular tool or molecular process for that. Okay, and then I think that they do an okay job in that YouTube video, which I think I put the link on iLearn as well. If you, I don't know if you can copy, you should be able to copy from the slides too, but that gives a little bit more of a video of how they do that, or how it works at least. Okay, and then the last one is Nanopore. Again, so these are kind of comparable, PacBio and Nanopore. They both are able to generate really long sequencing reads. And when we start talking about variation, we'll be able to talk about how how we use these tools to detect not just SNPs, not just single base pair changes, but whole changes, right? So what we're learning about, like in human genetics, we're learning that it's not just that the differences between someone who has a disease and doesn't is like a 1 base pair change, right? It's more than that sometimes. Sometimes it's a rearrangement. Maybe there's 2 copies of that gene that one person has versus another. Maybe there's a repetitive element that's near it, and we couldn't figure out where that repetitive element went until we had a long sequencing read. So to go back to my analogy of the puzzle, right, that you have a big puzzle, it has a lot of blue sky and clouds in it. If you use Nanopore or PacBio, the puzzle pieces are bigger, right? So you have more context when you're trying to build back a genome assembly with these, because you instead of having really small pieces, which is what I'm saying with Illumina, or small pieces, right? With Nanopore or PacBio, you get long pieces. As I said, some of these technologies can sequence 1 million bases in one run, which is pretty incredible. And so that allows the bioinformatics that we have to do to be easier, because it's easier to find that. Tanya asked whether or not, what's the error rate like? So one thing that's different between Illumina and is that both because you get so much data, because it's such a high, it's inexpensive and it generates so many sequence reads, there's a redundancy in it. So you can easily get deep coverage at a particular region of the genome. You can get 10 reads, that cover a particular spot. And so you're going to believe a variant. You're going to believe that something is actually an allele if you have a lot of things that confirm, a lot of reads that confirm it, right? So if I only saw one read that had a T and every other read had a C, right? And let's say there were 100 reads in that spot, right? In that one place, so I had 1 out of 100 said that there was a C there, but everybody else said there was a T. I would be inclined to think that was just a mistake, right? And so my computer program would say, you know what? If the allele frequency is like 1%, I'm not going to consider that a real variant. I'm going to think that that's a sequencing error, okay? But if I only sequenced 2 reads, right, and one of them was a C, one of them was a T, well, the ratio is 50% there, and without any other information, I might think that that's the right answer. That it's it's got there is a variant there, and there's 2 votes for that. One is a C, one is a T. So Illumina allows us to get deep coverage, and also because it's shorter reads and because of the way the technology works, the error rate tends to be pretty low anyways in the way that Illumina works. So I don't have the numbers here, and I don't want you to memorize them in terms of the actual numbers, but Illumina has higher sequence quality. PacBio tends to have lower sequence quality in the sense that any individual base tends, there's a bit higher rate of error in it estimating those bases. And the way they get around that is they still sequence the same molecule multiple times. So you can kind of overlap those together and try to come up with a consensus that that can throw out the kind of low performing bases or figure out what's the right base there. So, but in general, PacBio tends to have a higher error rate, as does Nanopore. So both of these technologies, while you can generate longer reads, just because of the way that they're working, right now there's still a higher error rate in them. So that's a disadvantage. Okay, so, and I will write this down a little bit. I think I'm going to add some more study guide questions for you to remember to come out of this, but Illumina gives you short reads, high quality, really deep sequence, because it's not very expensive. PacBio can give you really long reads. They tend to be, on an individual base pair, they tend to have lower quality, so it's a little bit less accurate, and it's more expensive to generate the sequence. Although I have to say, in the last year, it's gotten cheaper. So they've kind of, if you were to plot all these things, you'd see the curve coming down for that. So that's good. And then Nanopore is kind of in the middle in that it's got some high quality corrections, but it's still I would say on par with PacBio in terms of having lower quality individual confidence in sequence bases, but it can produce quite long ones, and it's probably on par with Nanopore with PacBio in terms of cost. So the cost is probably about the same. Okay, so oh, and I guess I didn't restate the chemistry, how the chemistry for Nanopore works, but this is the video of it, just to say that there's a little hole called a nanopore, called a pore, but it's very small, so it's called a nanopore, and a molecule can be pushed through that. In this case, it's a DNA molecule. It can be pushed through it, and there's a current. So that's what this little animation on the right-hand side is showing you. There's a electrical current, and what's happening is it's detecting changes in the electrical current as this molecule passes through. And if you train an algorithm on what does it look like when I pass an A through here, or more important, usually it's more like more than one. So it's like, what happens when I pass an ATC through there? And then it comes up with that signal. And so it does that enough that it learns how to train the algorithm on what it looks like, on what the pattern looks like in terms of the voltage change for a known set of sequences. And then it can work backwards later and try to decode the signal into sequences. And what's cool is, because it's just a pore, any kind of molecule that can pass through it works. So that can be DNA, that can be RNA, and now it can also use proteins. So if proteins are a little bit bigger, but it's still possible, and that's an ongoing, excuse me, research, is that people are able to, or research labs have been able to get protein sequencing working too. So, and then we'll talk a little bit more about epigenetics, but that's the idea that you would that the DNA molecule isn't just a DNA molecule. Sometimes those DNA molecules have modifications on them. One of those modifications is called methylation, and so if you were to train a tool on what methylated DNA looks like and what non-methylated DNA looks like, Nanopore can also distinguish between those. So there's a lot of advantages in some of these tools for that. Okay, so that's kind of from a size standpoint, like that's one of the, I wanted you to know kind of like how big the sequencing molecules, how big the molecules of sequence that these tools can sequence at a time are. And then I just, I guess, verbally told you a little bit more about the quality of those, the sequences that you get out. How am I doing? Does anybody have questions about, so I mentioned the genome size and complexity is kind of the other component of the lecture. Does anybody want me to go back over that too? Was that clear enough? Maybe I'll just go back to the first slide just in case that helps. And if it was clear, that's great. We don't have to prolong this, but I just want to make sure everybody feels like they understood. Let's see, for example, here, understood the ideas that were presented here, just that we can measure how much DNA there is in an organism, and before we could sequence the DNA, we could actually estimate how much DNA there was. And at one point, someone thought, oh, well, maybe the more DNA there is in a given organism, a haploid cell, maybe that means it's more complex. And these data are a way of saying, no, that's not true. So there's lots of things that are, quote unquote, I guess, less complex, like a small microbe, that have very big genomes. So it's not the case necessarily that. Okay. Okay, Claire's asked about the clone map and shotgun. I will happily go over that before I leave this slide. In case anybody wants me to do anything else on complexity, let me know. Cool. Okay, I'm going to jump over to the cloning. So I lecture on this because I think you should understand the background. The truth is that we don't have to do the we don't have to use clone maps as much, cloning and mapping, because of nanopore and long read sequencing. But let me just jump. I think this maybe visibly is still an important concept, and it's still something that we use. And if you take molecular biology, you definitely learn more about this kind of background. But the idea is that if we have the genome here as this total DNA that I'm drawing here on the left side, right? We break that DNA into pieces. And how do we break into pieces? Well, there's either a physical machine called a shear that shears it, that actually uses sonic waves to shear things, or there's enzymatic approaches where you can add an enzyme and it'll cut it at certain spots. But either way, we take the big DNA and we break it into smaller pieces, okay? And then we do what's called cloning, where we have this vector, right, which is basically this circular piece of DNA that can replicate within, and usually a bacterial cell. What we do is we can, there's essentially there's a chemistry and a biology that occurs that we can introduce these pieces of DNA, and they will be taken up by the cells. And because, you know, essentially organisms are trying to like fix problems all the time, right? Like, because naturally you could have an accident happen, right? There could be like an electric shock, or there could be even just like a pH difference that could cause something to be broken in a cell. You could have had a mutation or something that something that causes DNA to be broken. Cells try to fix that, and so they try to bring once they bring in this piece of DNA, if it's just sitting there, the cell's like, oh, they don't know the difference between DNA that was from their genome and DNA that came from somewhere else. So the cells try to fix it by essentially putting it back together. And so if you have this these vectors where they've been clipped, so they're open, and then you introduce this piece of DNA, naturally the cell tries to essentially fix it. And so that's what we take advantage of. But the point is that we're able to generate these clones that have small or large amounts of DNA from the that are pieces from the total DNA that came from the organism that we're interested in, right? So that's where we kind of are starting from. And the idea is that we can use these we can use that to essentially capture the entire DNA of an organism in what we call a library, where each one, a library is basically just in the original days was just like a growing E. coli cell that had in it a plasmid or vector that contained a part of the DNA of whatever it is we're interested in. So if it was human, right, there was a small fragment of DNA in there from a human. And so each one of those E. coli cells, and you could grow them in, big plates with 96 wells. I'm just trying to get to this slide. That in this clone here on the bottom left here, if here this, or it says array clones, right? We're fragmenting the DNA into these vectors. We're able to make sure that we're only keeping cell colonies where they have picked up this DNA from the host that we've tried to put in, and then now they're part of this living organism, and we can array them onto a big plate, and we call it an array, so we can use robotics to kind of move stuff around because it'll be systematically done. So the question is, how many of those little clones do you need to have a good chance of capturing the whole genome of your organism, right? And so you can use this math here to kind of come up with this estimate of how many you would need, right? And so really it's just a function of how big is the genome of the organism and how big is the fragment that you're putting in. So if you use a plasmid, which only can accept about a 2 kb, so 2,000 bases, and you had a human genome, then how many fragments would you need? And so that's kind of what we're showing there. I think I lost a 0 on my human. I gotta fix that for you guys. The human genome is a bit bigger than that. So, but unless I'm not sure why that would. Anyways, the human genome is 3,000 megabases. So maybe the salamander is bigger than that, and I didn't have it right. But anyways, the point being, depending on the size of your different organisms, you would need more clones to cover the whole genome. So that's kind of the like a strategy that was developed to come up with this. Again, the fact is we don't even have to do this anymore because we could just sequence the whole thing. But this was a strategy that people had to deal with before, when you had to sequence these basically 500 bases at a time that you would you would use to sequence a genome. The one thing I didn't really talk about was the mapping component of this, and I'm really not going to get into it, I think, in a lot of detail here, unless anyone's really curious about it. But because I'm not going to test you on it. But I think it's just the idea is that you have all those little clones. How could you figure out which chromosome they came from, so that you're not trying to sort of match this puzzle with everything, kind of where everything could be anywhere? And so people had developed other ways to take those E. coli cells that had the piece of DNA from the genome that was, I guess, typically a couple of hundred kb, right? So a couple 100,000 bases, and they can run restriction enzymes, which are another way to cut DNA, and then you can run that out on a gel, and you can see what that pattern looks like. And so basically, you can use that you can use it to say that two clones contain the same DNA if they had a similar pattern in this restriction enzyme mapping, basically. So, but since I don't have a slide showing you that, explaining it, I don't really want to. I'm afraid I'm going to probably confuse you if I try to just say it verbally. But I'm happy to provide some more references to that if people are curious about it. But I mostly want you just to understand the idea that we have to break the problem into smaller pieces. to sequence, and so we use that by doing this cloning strategy to take the big genome, put it into smaller plasmids or BACs, which are bacterial artificial chromosomes, or YACs, yeast artificial chromosomes. So basically like smaller than the whole genome, and then we can sequence those individuals and try to do that. But Illumina on the right-hand side is one strategy, doesn't even require that intermediate stage of putting things into plasmids because one, we have PCR, so we can amplify the stuff that we have, and two, we can sequence it so cheaply that it doesn't matter if we're sequencing something again and again, the costs are pretty low. So that's the kind of advantage there. All right, I'm gonna stop there for a second. Does anybody else have questions that I can help answer on the lecture? Great. Well, maybe we can figure out, it's really hard because I don't hear anybody's voices, so I don't know, and I can't see your faces saying yes or no on things, but let me just close this. I can see better. Yeah, if anybody has some feedback on if this is helping, or if you have further questions, I'm happy to answer them. Maybe it's coming across well enough in those lectures for now, and you're getting it. But as you start to study the materials and you do think about it, you're welcome to kind of write down questions and ask them on the discussion board or in other ways. Okay. I'm just going to wait a little bit of time in case anybody else is not feeling shy and wants to ask. Okay. A study guide that provides the key points to focus on. Yeah, I'm going to try to summarize that. So I think I'm trying to decide if it'll be a running document or if I'll just post one as we get towards the end of the of my section to remind you of it. I think but I will provide you a little bit more of a study guide with a summary of the key points. I tried to put a couple of those at the end of the lecture, but I can kind of give you a little bit more of an outline of that as we go. I guess I'll probably just go ahead and append those to the lectures that we've given so far, but it might be easier to have one document where you can just look at all of the summaries. So I'll see what's better for that. Yeah, no, I think so. Someone's chatting to me directly, just saying that they want to know what key points to study, and I think that's probably like I'm giving you a broad landscape, and you of course want to know what do you need to know, what do you need to take away from all of this, right? And I totally get that. I will give you a little bit more guidance about those key points, but if you can explain in some way what the different sequencing technologies have as advantages or disadvantages, that's helpful. If you can explain, what, how, if you understand how we can detect ploidy differences, then that's helpful. So some of these things are concepts more than just memorization. So I think that's what you want to maybe think about. Do you understand conceptually what's happening? We're a little constrained in the questions that we can ask you on an exam because it's only going to be the exam's going to take, it's only going to be as long as a course period. So it won't be, I won't be able to ask you a lot of really detailed essay questions or anything like that. So, but it'll be more about, do you understand the concepts that underlie that? So yeah, I will try to help you summarize that a little bit more. But sometimes I have to give you context for why we do this stuff. So that's why I kind of take a little bit of a detour. But cool. Okay. One more thing. Okay. So Cynthia asked about whether or not that the macro and micronucleus. Sure. And this is just an example of wacky biology. Is not. I mean, I'm happy to explain it. I'm not well, I might ask you a question about this on the test. We'll see. But the, oops, let me just share my screen. But this is, yeah. So this is an interesting example of crazy biology, and the reason why I think it was interesting, if you can see here, is when they, so imagine that you first sequenced the genome and everything was scrambled, right? That's what they found the first time they started doing this. They started looking at genes themselves. Is that what I mean by scrambled is, like, if you remember how a gene is organized, right? You have an ATG for a start codon, and it goes all the way up into a stop codon, right? And that makes a complete, what's called an open reading frame, and that has to translate into a protein. And we'll actually go over this again as we talk about mutations, but and how we detect them. But so somebody was doing this, they were sequencing a genome, they were putting it together, and they were finding that things were not in order, right? So if you put all those things, all those pieces together and you predict a gene, like none of the genes were really predicting correctly. And if you compared a gene that you would expect to find, so a gene like actin, which is a really important molecule in the cytoskeleton of most all organisms, that protein hasn't changed much. It's actually over billions of years. So the protein sequence of actin in a yeast is very, very similar to the protein that we find in humans or we find in plants. So when they looked at something like actin in one of these ciliates, they didn't find the protein, all the amino acids in the same order. They found things kind of shuffled. And so somebody had to figure out what was going on, and they realized through both microscopy and through some isolations of these different nuclei, basically by purifying them, that there were actually two types of nuclei in the same cell. And so what it is that one of those nuclei, the macronucleus, which is the nucleus where protein, where transcription is happening, right? Where the messenger RNAs are being made. If they just looked at those macronuclei, the genes, the pieces of the gene were in order. Okay, so that was consistent with what we'd expect. All the things that we know about biology were true there. Although when they started looking at the genome itself, they found that it was in 16,000 nanochromosomes, right? So basically so we only have 23 pairs of chromosomes, right? This had 16,000. And so they realized that what was happening was sort of almost like each gene or small fragments or multiple genes, but only in small fragments, were being put together. And when they were able to look at the micronucleus, this other component of the cell, when they looked for those corresponding places, they recognized that things were not in the same order. So these nanochromosomes were, the genes were in order, the components of the gene were in order, but they were in really, really small pieces. And in the micronucleus, they found chromosomes, so longer fragments, longer sort of contiguous sequence, but but the individual pieces of like the actin gene, the exons from the actin gene, weren't even in order in that genome. So that was crazy, and they had to figure out what happened. What is that led to that? And eventually they figured out that there's a molecular process that makes the macronucleus from the micronucleus, and there's basically a translation where all those little fragmented pieces, all those shuffled pieces get put back into the right order to make the macronucleus. So it's kind of like the blueprint. It's like, think about it this way. It's like you have the blueprints to your secret lair, right? And they're stored somewhere and they're shuffled, right? So not only are they're hidden in a small place, the micronucleus, but they're also encrypted, right? So they're all shuffled in random order so that nobody who ever got to read it, unless they knew something about how it was organized, could read it. Okay, and then in order to go have your henchmen make the lair somewhere on your island, right? You have to give them the actual blueprints, and so that's this translation of the macronucleus. So that then now everything is in order, but it's in really small pieces because of the way that the biology works there. So, anyways, it's just a crazy. I want to remind you that biology is pretty interesting if you haven't thought about it, and it's not just sort of what you might have learned in a genetics class of kind of like the classic, which is really important fundamentals, right? There's a classic idea between there's a genome, you produce a gene from that genome, or there's a gene in that genome, you produce a transcript, and that transcript produces a protein. That's great, but here's a case where there's actually kind of two genomes in these cells. Hope that. So sometimes I'm going to lecture about stuff just to make sure that you can get the breadth of what I think is kind of cool. And again, what's also important is none of this understanding, well, some of it could be done without genomics, but a lot of it required genomics to make sense of. And so again, kind of how the technology drives our understanding of the biology, I think is important. All right. The last thing I'm going to ask, well, I'll say something else, so then you can think about any other questions. Is so we're not doing like individual office hours in this, but some people might be feeling, you might be feeling shy, or you may not feel like you can ask a question in this forum since everybody's here. Um, reach out and let us know if you're being held back in some way by not feeling like you can ask. I mean, I think it's if we need to do something individually, or if we need to structure this time period such that we move into kind of like an office hours where there's kind of we can do what's called a waiting room, and then just one person at a time comes through, we can do that. But I feel like most of the questions everybody would like to hear the answer to, so I would prefer to do it this way. But in the same token, if you are having some individual problems, if your challenges at home, if there's something you need to tell me about that's about your situation, or and maybe it's not happening right now, but maybe it changes in the next 4 weeks. A lot of things might change in the next 4 weeks. You should reach out and let us know so that we can make sure that we can give you the best support so that you can still hopefully be able to learn this material. And so one thing I'm going to do is record my lectures over the weekend for the next week, so you can have a little bit more time to watch these and review it, so that in case your daily schedule is disrupted, you'll have a little bit more time to absorb this. Okay, so just let us know if anything else comes up. I'm not seeing any other questions, I don't think. All right, I'm gonna turn that off for a second. So yeah, I guess I'm gonna stop there. And we'll yeah, we'll pick it up on Tuesday, but you expect to see this weekend some posts for the lectures that are coming up. And I'll remind you about when the quiz is, and we'll get these kind of study notes. The quizzes will also give you a chance to kind of see the types of questions that we're going to ask. And hopefully it's making sense. And if it's not, I'd like you to let us know. Okay, great. I'm going to stop the recording now. And